good education from a regional language, that is Kannada language. He has come up, and he has featured also on many TV interviews for his work, and he was a panelist for the interviews that are featured in on BBC also. With this short biography, I would like to welcome Dr. Rajshekhar Giraddi for his talk. Sir, the platform is over to you.
Thank you, Dr. Bharati, for your kind introduction. I'm really happy to be presenting some of the notes that I have in my mind to all of you. I would like to begin with an apology because I believe that all of you would be engineering students without a biology background. So I've kept the talk very simple, which might help some of you. But for those who need more experimental details and technical help, please feel free to reach out to me. I'd be more than happy to discuss it further. So with that, I'll try and present it with my slides here and see if that works. Okay, let's see. Are you guys able to see my screen as a full screen slide?
Yeah, it's good.
Yes, sir, it's coming.
Okay, excellent. And I have already assumed that you guys can hear me, so I'll begin the talk. Hi, again, my name is Raj. I'm a senior scientist doing cancer research in Pfizer in California, USA. Today, I will briefly talk about some of the scientific concepts that I would like to share with you briefly on coronavirus and cancer research, and then end the talk with some of my views on career goals and prospects, as requested by some of you. So the agenda for today's talk would be a brief introduction to coronavirus and why it was so easy to win a battle against this disease. And then I'll give an introduction to cancer and then explain why we're still struggling with cancer. And then I'll end the talk with some of my views as an average student and what helped me to be successful in my profession. So I'll begin with coronavirus. You guys have heard this many times, but I'm just going to give a bit of scientific prospect of what this virus is. So what is coronavirus? It is a virus, and there are thousands and thousands of viruses around the planet, and you would have been infected by a lot of them. And this particular virus and these thousands of viruses have all been scientifically organized into different families and subfamilies based on their features and properties. This particular coronavirus belongs to a family called SARS-CoV-2 family. Some of the viruses in this family affect humans, and it's likely that some of you or almost all of you have already been affected by some of the viruses from this family, but most of them don't really have a drastic symptoms or a phenotype, and that's why you may have just noticed cold or just a flu-like symptom and you ignored it. But it is likely that you've already been infected by some of these viruses. There are some viruses in this family, they only affect animals and they don't affect humans. But in rare cases, there are some viruses that jump from animal and then start infecting humans. Now, there is a theory that this coronavirus is one of that kind, which was actually infecting animals earlier. It picked up a mutation, and then it's now starting to affect humans. And so that's why the scientists came up with this new virus and gave this scientific term. You may have heard this before, COVID-19. It stands for Coronavirus Disease Identified in the year 2019. So what happens if you're typically or unfortunately infected by this virus is that you may get some of these symptoms or all of these symptoms. Most of them will resemble a similar cold or flu-like infection, such as fever, cough, sore throat, or congestion. But some of the symptoms will progress further, and you'll have difficulty in breathing, and then you will also develop other complex symptoms. Some people are able to recover from this easily, but some people have a deadly symptoms, and it ends up being a very serious infection. Now, why? Now, you are actually infected by a virus on a daily basis. It's likely that you're being infected by a virus as we speak now. And it's a common process, and our body knows how to fight a virus, and most of the time we don't notice it and we ignore it. But there's a reason why coronavirus became such a deadly disease. For a virus to go deadly and become a pandemic, it needs two things. One is its infection rate, meaning how good it is in affecting and spreading one patient to another patient to another patient. And then once it infects someone, what kind of impact it has on patients? Can we easily recover from it? Or does it lead to serious symptoms that require hospitalization? Now, you might wonder that for a coronavirus to be pandemic, that this was super infectious and this was super deadly. It's actually the opposite. If you have a virus that is very, very infectious, that means it's actually easy to catch it, and people will realize that this infection is happening quickly and it is easily contained. So the reason why corona became successful is because it was not very good at infections, but it wasn't bad at infections either. So people did not realize it, and we sort of ignored it in the first few months. The second thing is deadliness. If it is too deadly, then patients will realize if two people are infected and if both of them die, then it'll be a major news everywhere. And people will immediately notice it, and it'll be contained to a very, very small geographical location. But what happens with corona is that some people have no symptoms and some people have deadly symptoms. And that's why even when you're infected, you would have ignored it. So it sort of came in that middle spot. And that's the reason why coronavirus became such a big pandemic. So I have a graph that explains how this actually works. So in this graph, a number of viruses have been plotted on this graph. On this axis, you have the infection rate. On this axis, you have the impact rate. So you will see something called as the common cold that we always get. It's very difficult to get infected, and it's actually very difficult to have an impactful disease. So we sort of ignore this common cold. You may have heard of something called this chicken pox, and this is a very, very easy to infect virus. It gets transferred from one patient to another patient very, very easily, but it doesn't have any deadly symptoms, and that's why people sort of ignored this one as well. On the other spectrum, you probably heard of something called as Ebola, and that is a very, very deadly disease, meaning that if you get this Ebola virus, it's likely that you will die because its fatality rate is almost more than 50%. But because it was so severe and dramatic, people noticed it very early on, and it was contained to only few countries. But what happened with coronavirus was that it was not too deadly and it was not too infectious. So people ignored it and they didn't realize what was happening. So coronavirus fell in this sweet spot and ended up becoming a pandemic. Another thing you will notice in this graph is there's a virus called SARS. SARS was also a pandemic a decade ago, but it was not as popular as coronavirus. But the biggest reason is that, in fact, SARS and coronavirus are quite similar to each other in terms of infection and impact rate. But what happened at the time of SARS was that there were only 1 billion people who were traveling on a daily basis. But in today's time, there were about 4 billion people traveling around the world, especially traveling between the country. So the spread was quicker and it spread a lot more to different countries geographically. And that's why in everyone's memory, Corona became the biggest pandemic that we would ever know. But there was one good thing about it, meaning that it's a virus that we all know where it comes from. So all we had to do was come up with a vaccine for it. Now, most of you may know how vaccines work, but just for the sake of safety, I decided to just use a simple explanation of how these vaccines work. So what are vaccines? So vaccines are actually just a weak form of the virus, or it's a dead form of the virus, meaning that we are actually injecting some of the virus into your body, but in a weaker form or a dead copy of that virus. Or quite often, we only inject half of the coronavirus and we leave the other half out. And that's quite often how the vaccines work. So when we inject these weak, dead, or half of the viruses, then our body reacts to it. And we have immune cells in our body that immediately go and attack these. And because these are weak or dead, they don't have the capacity to infect, but they're still good enough to generate an immune response. So what happens that some of the cells called B cells and lymphocytes, they will recognize it and they'll remember that these are, in this case, this is a coronavirus, and then we need to know how to attack it and then remember it. So this is surprising and amazing to know that our body keeps a central database of every single infection you ever had in your entire life. So if we give a weak form of the corona vaccine, and if it is trained enough to handle it, then when a real corona vaccine is infected, then it'll immediately know its antibody database. it'll pop up that antibody, makes it in millions, and attacks the virus before the virus has a chance to affect us. So that's how quite often the vaccines work. So because we knew something about the coronavirus to begin with, it was quite easy to come up with a vaccine. And the way we went about is I thought I would just give a brief description of the structure of the coronavirus to let you know how that works. So this is an example of a microscopic copy of a coronavirus. And the reason why it's called corona, meaning that you see these pink colored proteins. So these are different types of proteins. And under the microscope, they appear like a crown, the one that you'd keep on a king's or queen's head. So they appear like a crown, and the crown is what we call as corona in certain language. So this is an example of how the virus looks. And when you split open this virus and look inside, it is made up of a number of proteins and number of copies. All of these proteins and components are very, very essential for a virus to go recognize a human cell and infect them. The best thing about the coronavirus and why it became easy was that all of these proteins that we see here will always remain quite similar. They do not mutate, they do not change, and it's very easy to understand them. So we knew a lot about these proteins, and what Pfizer did is that instead of looking at different proteins here, it simply worked on this one protein called the spike-glycoprotein. So we took some of the sequences from this glycoprotein, designed an RNA-based vaccine. We ended up testing it in few patients, and then immediately launched into a global clinical trial. So because it was so easy to study this virus, and because this virus does not mutate, and because Pfizer had the infrastructure and previous knowledge of working with vaccine, we had a record time of just 10 months to know a virus and then come up with a vaccine as a treatment for it. Something like this has been impossible for other viruses such as AIDS or HIV, because all the components that you see here, the different proteins, they keep changing. So if you design a vaccine for one protein, before you know, that protein has changed in the next week. So the vaccine is no longer effective. And that's the reason why some of the viruses are very difficult to come up with a vaccine. But we had been lucky with coronavirus, and that's how we ended up winning it. It sounds like it was a big problem, but from the scientist perspective, it was actually a very easy win to come up with a treatment for this. So that's all the things I had to say about the coronavirus. So with that, I'll move on to cancer. Again, I apologize if most of my talk has been less scientific, but let me know if you need more details. Now I'll give a brief introduction to what cancer is. And unlike coronavirus, we are still struggling with it because coronavirus was only detected 1 year ago and we already have a treatment. But cancer was detected more than 100 years ago, and we still do not have any treatment for this that is very, very effective as a vaccine as coronavirus. I'll just give a brief introduction to cancer and then explain why the problem is so complex for cancer. I'll begin with what cancer is. What cancer is, in scientific term, is uncontrolled cell division. Now, you may or may not know what a cell division is. I'll briefly go through that. Our body is made up of billions and billions of cells. And on our daily basis, we walk, we eat, we run, we exercise, our hair grows, our nail grows, we have wounds that need to be healed, our lungs are damaged, our liver is damaged. So in all of those processes, there's some sort of regeneration that's happening all the time in our body. And the way that regeneration happens in normal circumstances is that you have these cells, and when they detect a problem or when there's a need for any healing or regeneration, they divide. They know when to start the division, and then they'll stop when they have achieved. For example, if a wound is healed in a certain location, they'll stop dividing. They'll say, okay, the wound is now healed. We don't need to divide anymore. What happens in cancer is that you will have some of these cells that are dividing, but they unfortunately don't know how to stop their cell division. So they'll just continue to divide and divide and divide. So this uncontrolled cell division is what we actually call as cancer. People don't know, no one actually dies from this uncontrolled cell division because in reality, this happens at least once or twice a day in everyone's life, every day. But our body knows how to detect these cancer cells, contain them, kill them, and get back to the normalcy that is represented on the left side of the picture here. This happens on a daily basis, but over the time, this doesn't work. And quite often what happens in those cases is that these uncontrolled cells will overtake everything else. They'll invade, they'll extend into the normal tissue, they'll change the structure of our organs, they'll go to the next organ and the next organs. So because of those complications, a patient will start accumulating different ailments. And what happens at the end is that patients actually die because of ailments that come as a result of this uncontrolled cell division. No one dies from this uncontrolled cell divisions, but they die indirectly because of the problems it creates, which is easily explainable. For example, when you have a patient with a liver cancer, one patient with a liver cancer may die because their liver has stopped working. Another patient with liver cancer may die because their lung has stopped working or their kidney has stopped working. So when you actually look at it at the end of the day, no single patient dies for the same reason as a cancer patient, but because the cancer affects different people in different tissues in a very, very different way. So that's the gist of all. Now you may want to know why this occurs in our body. Why do we get cancer and how does it begin? What happens in normal cells, for a cell to know when to start and for a cell to know when to stop, there are a certain set of instructions provided by a list of genes. I hope you all are aware with the term gene. These are instructions of what happens with our cells and how to regulate everything in our body. So these instructions then come in something called as genes. So for every action that happens in our body, there are at least a basket of genes that are regulating it. These genes will tell you when they should divide and when they should stop. But what happens over the age, changes in the physiology, sometimes purely by chance or accident, and some other environmental factors, what happens is that at least just one of the gene gets mutated, as represented in the red color here. Only one of them gets mutated and becomes a different type of protein. And again, this happens on a daily basis in every person, but our cell and body knows how to immediately fix it and repair it. They usually take this protein and remove it out of the cell and destroy it. But quite often that doesn't happen. Sometimes the cell let a damaged protein survive in the cell, and this damaged protein or the mutated protein will now start affecting other proteins and other functions in the body. And what happens over the time is that this goes on and on and on. Now you'll end up with cells which have lots of different types of mutated proteins. They really don't know how to behave. They don't know how to regulate, but all of them are channeling uncontrolled cell divisions. And that ends up with lots of cells being produced, almost billions of cells being produced on a daily basis. Now, you may appear that that's the end of the story with, starting from one protein with 4 proteins being mutated and affecting everything. But what happens in reality in cancer patients is that it's an entire cascade. Here is just one small snapshot of so many proteins going wrong, so many cross-linkings happening, different interactions. So what we do as scientists, as a cancer research scientist, is that we try and dig through this information to see if any of that is important for us to come up with a drug. Now, for example, here it looks very clear that this AKT protein, it's in the center of everything. So you may think that that's a good place to start and design a drug for it. But before you know, the cells will undergo mutation and they'll develop a cross-link to the point that they no longer need AKT protein, but they're still able to proliferate uncontrollably. So that's the same thing happens no matter whenever you come up with a drug. Quite often, we come up with a drug that's very effective, for example, EGF. When you design a drug for this protein, that's usually affected and cancer cells die. But what happens in that process is that EGF is very important for a lot of normal tissues. Some of the other normal tissues stop functioning, so the patient will actually die of consequence because of the side effects and the cytotoxicity. So there are very rare cases where treatments using something like an EGF may work, but in reality, what is happening in every cancer cell, in every cancer patient, is that lots of these proteins are being detected and they're being mutated and they're developing all sorts of crosstalks and crosslinks, and they're just letting the cancer cells divide again and again. And that's the reason why cancer is so difficult to treat. And it's compared to the coronavirus, where you have at least 5 or 6 proteins, they don't change, they always behave the same way. So as a scientist, corona is a very good boy for us to work with. but something such as cancer, it's very, very difficult. So what I do in Pfizer, what my colleagues do in Pfizer on a daily basis is that we still haven't finished digging through all of this information. So on a daily basis, we look at different genes in different cancer samples and see if any of that makes sense, if one protein is dominant over the other, or if one is upstream or if one is downstream. So we are coming up with different iterations, different permutation and combination, and then see if any of these proteins can be used as a therapy against cancer. And that's what we do. But not everything is bad news for cancer. In fact, we have done remarkably well when it comes to treating cancer patients. So now you have a number of cancer treatment options available. The first one being precision medicines, where you can come up with a mutation in a patient and then actually get lucky with a good therapy that only attacks that mutation. The best example for a precision medicine is a subset of breast cancer patients. And there's a defined mutations and there are drugs available for breast cancer. And once you're done with that treatment, it's likely that you don't get the cancer again. And we know hundreds of examples where women have never got breast cancer, even stopping cancer treatment after 20 or 30 years, they still don't have any cancer coming back again. The same thing with some of the cancers in children. Some of these can be easily targeted by a mutation, and there are medicines available for those cancers. And once the cancer is gone in those kids, in adulthood, it does not come back. So we do have some perfect examples that some of these are actually working, but we haven't reached to a final story yet. We have also made a lot of improvements in precision and the way we do our surgery using robotics and using clear margins. So we are able to actually detect it very early and remove before it gets bigger or before it goes into other tissues or organs. Our chemotherapy has also changed dramatically. So now it's less toxic and patients are able to tolerate. And we now know exactly when to give and when not to give. There's a new branch of medicine coming up called as the immunotherapy, where scientists are coming up with ways where our immune cells are trained how to kill cancer cells. And this has been the recent discovery. And in fact, a year or 2 years ago, this won the Nobel Prize because it has already affected a number of cancer patients. And this has been a successful therapy for thousands and thousands of cancer patients. So a lot of good things are going on for cancer treatments. In fact, when you look at the death rate, and here I'm just giving an example, 4 different types of cancer, but the same story goes for a lot of different cancers. When you look at ovarian cancer, for example, in the next 10 years or so, we're going to reduce the death rate by 40%. And that's very remarkable compared to what used to happen just in 1990s. And the same story for breast cancer, bowel cancer, and prostate cancer. So we are continuing to win our battle against cancer, but it's not the end of it, and we're still getting there. But there's one thing I wanted to mention today, and there's a lot of misconception about cancer, and I thought that I should use this platform to just briefly discuss that misconception and give you a take-home message. So I think most of you agree that nowadays everyone knows somebody or the other person in the family who's got cancer or who has died of disease. It has become a very common disease for all of us. And quite often the misconception is that, I think the cancer has become common because our lifestyle has changed dramatically, or something has happened in the last 20 years. We're looking at mobiles more frequently, or we're looking at laptops more frequently, or we have stopped eating roti and we're eating pizzas and burgers. And there's a lot of misconception that our lifestyle and diet behaviors have changed, and they think that that's the reason why now cancer has become so common. But the real story is that the number of people getting cancer has never changed in the last 50 or 60 years. But the reason why you hear more cancer patients is that there's just literally more people on the planet right now. So 20 or 30 years ago, we had a very different population. Now we are about 7 billion people. So naturally, for example, even when you look at the number of people who have headaches or the number of people who have fever, you just have too many people who have fever or headaches. It's just because the population has exploded. So the same story is for cancer. None of our diet or lifestyle has suddenly affected the number of cancer patients that we are observing around. It's just that we have a lot more people. The second reason why we are getting more cancer patients, if any, we are getting a little more cancer patients these days, is that biologically and classically, cancer has always been associated as an old age disease, meaning that if you're less than 70 years old, you are 40 times less likely to die from cancer, meaning that as soon as you cross 70 and beyond, your chances of getting cancer or dying from it actually increases. What used to happen in the past that people simply died before they became 70. People would have died at 40 years old, 50 or 60 years age because of infections or plague or less health infrastructure. But now with clinical infrastructure, improved diagnosis, and number of clinics available around the world, people are actually living longer. And because they're living longer, they're reaching that dead point where they might actually end up getting cancer. So it's the combination of increased population and the combination that we are actually living longer is why now you hear the word cancer very, very often. So just keep those in mind. It's got nothing to do with diet or lifestyle. There's no misconception. The only direct link we have had so far with our lifestyle is smoking. Apart from that, nothing else seems to be directly correlated to the incidence of cancer that we get. So keep in mind that that the cancer care is working and the death rates are falling. And we at Pfizer, or the scientists around the world in multiple universities in different countries, are working very, very hard to come up with a sort of a good solution that will work with a lot of cancer patients. So with that, I'll finish my science talk and then move on to the last bit and give you some advice because it was suggested that I should do that. I've always been an average student, so all my suggestions and views will come as an average student. I've never been a smart person. So I'll just go through what I learned as a student and how that changed over the time so that you have the benefit of learning in advance. As a student, I always learned that there's this unintentional hierarchy in our society. At any time, whether it's a classroom or a college or a society or a country, you can always divide it into different hierarchies. There's a top tier, and there's a medium tier and an average tier. And this sort of hierarchy works in multiple ways. For example, if you just look at people with money, you will have three hierarchies. So you will have people at the top with lots and lots of money. And you will have people in the medium with decent amount of money, but they could make more. And people at the average level, they have less money and they really need to make more. And similarly, not just the amount of money, when you look at someone's professional life and someone how successful they have been in getting job and being successful in their career, there's again a hierarchy. There are people who work in companies such as Google and Apple, and there are people who work in Infosys, Wipro, and there are people who work in lesser known companies. So people always put these companies into different hierarchies, and they will also associate people working in those companies into these different hierarchies. Quite often, even if you just take a simple classroom, even in one classroom, you can come up with this sort of hierarchy where you have at the top, you have few students who are teacher's favorite, they score very well, and it's very easy for them to land a job. And then you have medium students, they're quite regular students, they score well, and they'll eventually get a job. And they will also have some regular students, they have average scores, and they find it difficult to come up with a job, and quite often you call them as a backbenchers. So I hope you all agree that this sort of hierarchy works in most of the places around the world. So when I was a student, I really believed that this hierarchy exists because this is what I witnessed in my classroom. I saw that there were three classes of people into these categories, and I really thought that's the hierarchy that exists everywhere. And what I also thought was that this hierarchy of top scoring students, regular students, and average students, it has a direct correlation to their smartness level. I always believe that people who score well, who are always at the top, are super smart people. They have good IQ. I always believe that the medium people are okay-ish and dumb people have always been average. I somehow had this notion because I was never a smart person. I was always being a dumb person. So I just assumed that as a dumb person, I'll always end up in an average category and ever reach top of my profession. And that's the kind of notion I had. But even while I was a student, I quickly realized that that is completely wrong because I met a lot of super smart people in my own classroom. And when I asked them what they want to do with their smartness and what they want to do with their life, they really had no idea. I also met some dumb people who were really not good in textbooks, but they had some of the smartest ideas of what to do in their life and how to be successful. So I realized that none of this hierarchy exists, and it has no relation with intelligence level. So I quickly changed myself. Instead of relating my success to whether I'm dumb or smart, whether I'm going to be successful or not. One thing I changed in my life was to immediately, even as a young student, I decided what to do. And just to give an example, and this is the only thing that helped me, I quickly decided what I wanted to do in my life, and I just drove right into it. It's like, you know what, this is what I want to do, and I'm going to work on it. So in 2004, I decided that, hey, I'm going to be a scientist. And that's one of the reasons I never prepared for exams. That didn't help me to be a scientist. So when I say dumb in the last slide here, I can give you an example that in my PUC, my score was about 62 or 63%. When CET rankings were announced, my ranking was beyond 25,000. And I'm talking about 2000 decade. So CET ranking beyond 25,000 means that you pretty much didn't have a chance to get an engineering seat in any college in the state. But the reason I got 25,000 ranking is because I didn't study for CET. When everyone else was preparing for CET exam, I was in Bangalore doing my research on different biotechnology classes, different microbiology classes, assess which college is good, which is going to give me a good prospect for cancer research. And that's the kind of example I wanted to put out here, is that as a very, very early on in your student life, you have to decide what you want to do and then just start acting on it. Throughout this time in 2004 or 2006 or 2008, I was never brilliant. When I decided to be a scientist, I was a very average student. I never got any good marks. When I started my first job as a scientist, I was really not good at my job, but I knew that I had to get there. So I just kept working on my goals. And that eventually paid off for me. And in 2008, I was selected to do a PhD in Cambridge University. And from that time onwards, I just built my professional life. So my only two cents of advice for you guys as a student is just decide what you want to be. It doesn't matter what you decide. If you choose to stay in your own city after college, get a job in a company that you like, and then just act on it. If you choose that, no, I wanted to be in Google, then you have to just act on it. You just have to decide what you want to be and own your decisions and just start acting on it. Because I think only students have the strength to start acting on what they decide, because as you grow older, that strength to act on it, or in the mid 30s and mid 40s to have a different turnaround in your life or to come up with a different decision is very, very difficult. But I think only students, in my experience, have the strength to decide what they want to be. And that has always been the case in my life, even with my friends and colleagues around the world. Those who are in the position as same as me have been the people who decided very early on what they wanted to do. We didn't really put extra hours in our classes. We didn't study more than other students, but we just chose to spend our time correctly on what we want to do. Another example I could give is that I had a lot of friends who were, preparing for exams because they wanted to be the first rank or second rank students. But I choose not to do that. Instead, I would go around IIC or other research institution just to get a glimpse of what research is and what kind of experiments they're doing, and just get an idea of what is required to get into those places. So once you decide, it's actually very easy to get and reach where you want to be. So if your destinations are higher, but if you decide earlier, I am pretty much guaranteed that you will definitely reach there. And that's all I wanted to say today. And with that, I'd like to thank for all of your time. And I really thank for your patience for sitting through the entire talk. If you have any questions about the scientific knowledge that I just mentioned, or if you have any advice on career, you can reach out to me on that email ID. I'd be more than happy to help you when it's possible. Thank you.
Thank you for your time, sir. We actually have a few questions on from YouTube and the meeting. So Gautam and I will be asking you those. First question, is the vaccine of coronavirus come to India? If not, when will it come?
It will come to India. It's just a matter of logistics. I believe that some of them are already being shipped, so it's not very far away. I think in the month of January, you will probably start getting some of those vaccines.
Okay. Mr. Biku Bave asks, many people are working on Desi cow urine therapy and Panchayagna therapy for treating cancer. Is there any research going on in this?
Do you mean natural treatments conditions for
Yes,
cancer?
yes, by the looks of it, yeah.
Um, there's a lot of research going on in that, but some of the one of the reason why it's very difficult to publish those and come up with an accurate prediction is because we don't have an active chemical matter to study. So when you're giving such treatments, we don't know what gene is being affected because of what reason. So when you want to explain a molecular mechanism of how it affects, it's very difficult to prove it. And that's the reason why no one does those experiments. But the alternative way to prove if it's effective is to do it on a very, very large scale basis. But I don't believe there's any government proposal where they're thinking of taking thousands and thousands of cancer patients and subjecting them to natural therapies. So I think there's a point that this may never be proved because of the way these studies are conducted, but there might be some benefit that we are probably ignoring there.
Gautam has a question for you now.
So over to me. So we have a question from the HOD of AI/ML department. So she's asked, could you highlight on how Pfizer identifies the role of radiogenomics towards identifying digital biomarkers and their significance in prognosis, diagnostics, and therapeutics in oncology?
Yeah, that's a wonderful question. I mean, it's not just Pfizer. I think these days, pretty much every other industry uses these tools. And quite often for me, on a daily basis, I use these tools because I think if you guys recall from a picture that I showed earlier where, one protein is interacting with, 20 or 30 other proteins, it's not very easy to predict manually these days. So some of these new computational tools and techniques have been very valuable to come up with interactions of these proteins and make our research go faster. Another way it has helped us is to, as you mentioned, is analyze large amounts of data quickly and identify biomarkers that are easily reproducible. So again, that's another place where it has helped significantly to most of the universities and industry. And lastly, there are millions and millions of chemicals, and we have never studied if any of those can actually be a good drug candidates for cancer. But now, the reason why we don't do that is because it's simply impossible to take millions of chemicals and then test them against millions of cancer cells. So what artificial learning and some of these computational tools are helping is simulating some of the potential interactions in the computer and then give you a short list of chemicals that you want to test further. So we certainly use these on a regular basis, and it's not just Pfizer.
So I also have a question for you. I just want to know how the impact of AI has been on drug discovery.
Remarkable, and pretty much now most of the industries will collapse without AI. Because with the increased technology, AI being available, there's increased demand to run the research programs run faster. There's always a competing company using these tools. So if you choose not to use AI in your cancer research, then you'll be working slowly and lagging behind against other companies and labs. So pretty much it has become an essential tool for us.
Yeah, thank you, sir. We also have another question. So could you highlight on the opportunities that engineering students have in biopharma research companies like Pfizer?
Right, yeah. So there are actually two different ways engineering have an impact on healthcare and research. Actually, Pfizer employs hundreds and hundreds of engineers. And the reason is because, for example, you can think of coronavirus vaccine. Those require large-scale engineering plans to come up with bioreactors that can make millions and millions of vaccines. And as biologists, we have no clue how to design those and construct those and how to run those. So computational level, civil level, architect level, we really need a lot of engineers, and Pfizer on a regular basis hires a lot of engineers. So that's one place where you would be of impact you would have an impact. The second is, as I mentioned before, as computer engineering students, nowadays it has become very common for IT students to learn a little more about the biology and use some of these machine learning tools and big data set tool to actually analyze cancer patients. And on a regular basis, we get applications from IT people looking into that. So, if you ever pick up biology in the future, there's a good way to go. In fact, know someone who's now running an entire program on cancer research, and for the last 20 years, that person was an IT person in Microsoft. So it's very much, interlinked these days.
Thank you, sir. So with so many vaccines suddenly entering into the market, so how would a common person decide which one to get for himself?
Quite often, it's unfortunate that as patients, we may or may not get to decide what vaccine we're going to get, because that all comes down to your government. The kind of documentation and the collaboration the government has done. For example, government of India has preferably chosen the one that's being made in Pune, and it's likely that you have a higher chance of getting that vaccine compared to the one being made by Pfizer or Moderna. It just comes down to logistics, pricing. But on a scientific level, all of these vaccines should pass the threshold. And once they are approved by regulatory agencies, it means that most of these vaccines are actually pretty good candidates. It's just logistics and your local restrictions that will help you decide which vaccine you will end up taking. For example, there's a debate going on right now on whether some of the rich people in India can access Pfizer vaccine, because now that's the most expensive. Indian government is choosing not to act on it because it's very expensive for it to run a large vaccination for billions of people. But there's a certain class of rich people that say, hey, we don't mind paying the price. If it's ready, we'll take it next week. So it really comes down to your government regulations, approvals. But if a vaccine says it's a vaccine, it should work. It doesn't matter which brand it comes from.
So next question. Is the virus a natural virus or a man-made virus? Can you elaborate on its origin?
We cannot elaborate on that because those studies are going right now. In fact, just this week, an expert of 10 scientists from, I believe, World Health Organization have landed in China to understand what happened. It's likely, given the structure and the nature of that protein and the way it has been studied by multiple independent scientific organizations, it's highly likely that it's a natural virus. It's very unlikely to be an artificial virus made in lab, because everything points out to being a natural organism. But we just simply don't know how it jumped from an animal infecting virus to a human infecting virus. It has happened in the past. We know how that works in other viruses. We simply don't know how it happened in China for this particular virus. So the study has been mostly focusing on the origin of virus, not understanding whether it's artificial or natural.
Okay, the same person has asked this question. There are many reports about a perfect vaccine for the virus. Is there a possibility of getting a perfect vaccine?
You will not have a perfect vaccine for all viruses in the world because, as I mentioned before, thousands of these viruses have been classified into different families and subfamilies. And it's likely that you can come up with a vaccine that affects the members of everyone in that family. And you will have another vaccine that affects every other virus in that family. There's a good chance and potential in the future that might be the case, that you will have a vaccine for each family, but it's highly unlikely that you may come up with a vaccine that is universal for all viruses, because all viruses have different structures, they behave differently, they regulate differently, and some of them mutate themselves, some of them don't mutate. So it's just different permutation and combinations. Unlike, for example, bacteria, most of the bacteria have some of the common elements, and that's why no matter which bacteria you're infected with, if you give them an antibiotic, you're likely to recover from it.
Okay, we have a question from a student of our department. How does supercomputers such as IBM Watson help doctors and researchers in curing cancer and understanding it? Do they prove to be useful, or is it any better?
No, very, very, very useful. In fact, I have my own private login with a supercomputer that I often use at UCSD. And if I don't log in and use the supercomputer, I will not be able to analyze my data. And the reason is because in these days, we end up with an Excel sheet that has a million rows and million columns. If I use a simple computer with an Excel sheet that has a million rows and million columns, it'll simply crash. So, in fact, most of these days, we all need a working computer, and not everyone will need an IBM supercomputer, but we certainly need supercomputers. And IBM has its own specific research program, and it has its own way of helping cancer patients and cancer research. And I think there's a good example of that it's actually doing a good job.
This will be a question, the last question I'll be asking. So from the next question, anyone who has a question can unmute themselves and ask one by one. So Ahmed is asking, what do you mean by strains of COVID-19? Will the vaccine be effective against all strains of the COVID-19?
That's a good question. So when a virus starts infecting people, it's going to interact with different hosts, meaning it's going to interact with people with certain genes, and different racial people have different genes. So quite often, based on the patient that's interacting with or infecting with, you may end up coming up with varieties of that strain, and you will have more and more strains being developed. So for this one particular example for coronavirus, we currently have at least 5 or 6 known strains of coronavirus. All of them seem to be affecting patients across the world. But the vaccine that we have, and the vaccine that most people are working, is on that spike glycoprotein that seems to be the common variant for all of these strains. So in our research, it has shown that the vaccine is very effective on all the strains.
Okay, I'll be continuing with the questions on chat.
Yeah.
Sorry?
Yeah, go ahead. Go ahead.
Okay. How can a user decide on the vaccine? Is it just by watching for one of them with the least side effects reported? Or how do you exactly measure the efficiency of a vaccine from a common man's perspective, not from, say, the government or anything?
I know. So only 2 parameters that work. So one is how effective it is. So meaning that when you get a vaccine, and unfortunately, if you're infected in the future, is your body going to be good enough to know from the previous experience of the vaccine? And the second thing, as a common man, of course, is side infection. So when side of infection, such as, are you guaranteed to have a fever on the day you get vaccine? Or there's a very slim chance that you get fever on the day you get vaccine. And that's sort of, common person's question about a vaccine. These questions are quite often answered by the regulatory agencies before it gets approved. So if you come up with a vaccine that's likely to induce fever on the day of the vaccine, then it's likely to be not approved by the government. And it takes longer for the companies to refine it further and then minimize the side effect. There might also be some allergic reactions, and they're also studied in large clinical trials. So what I would tend to say for common people is that if a vaccine has gone through scientific review and has been approved as a good vaccine, then I would say simply just take it.
We have a question from YouTube now. With the vaccine coming out, how long is it going to take for things to get back to normal? Since we're wearing masks and trying to follow social distancing, how long will we have to continue after getting vaccinated?
So we can't speak for the exact deadlines and timelines, because we cannot predict. The reason why we can't do is because we cannot predict the traveling behavior across the globe. But if everything is going well, and if most people are getting the vaccine as they need it, we will be end of this coronavirus pandemic by the mid of next year or by the end of next year. It all comes down to how quickly people are getting the vaccination. And it's not critical that every single person should get the vaccination for the coronavirus to be effective, for the pandemic to be over. Even if half the people in this world get vaccinated, that is already ending the pandemic. So I think we are at least 6 months before the end of this pandemic, but worst case scenario by the end of next year. But the social distancing protocols and the mask will continue to follow for the rest of the year.
So another question: what are the common side effects of the vaccine that's currently there?
Right. Often, for most people, it's just fever and cold-like symptoms or headache. But there are subsection of people, and we will never know, but we would have known earlier as a child when the child has gone through other vaccination programs. There are very, very rare subsets of people who have allergic reactions and hyperactivated immune response. And you would have known that as a kid. So if you would known that as a kid, it would be in your medical records. So before they give you a COVID vaccine, they'll ask you if you have been allergic to any other vaccine in the past, like in it as a kid. So with that, you will know. But otherwise, a common but a rare side effect would be fever and muscle pain and body pain.
So the next question is, if the virus mutates, and how effective would the vaccine be?
So I think so far, most of our studies have led to the conclusion that this particular virus does not mutate rapidly. So with that confidence, I think the vaccine is going to be super successful. In fact, as I mentioned earlier, it has gone through about 5 or 6 iteration of mutations, but all those mutations have been preventable with one vaccine that has been produced by multiple companies. So I think this particular virus, the mutation is not going to be a big problem.
Another question: as a part of Pfizer, would you volunteer to take a vaccine shot?
Oh, I would be more than happy to jump in because I believe in the scientific strengths and the merits of the kind of research that we do. But I think, as I just mentioned, as it was mentioned by a CEO of our company, just a couple of days in the news, that we strongly believe in the quality of science we do, but we cannot cut lines. And I think there are doctors and nurses and old people who need this vaccine more desperately than we need. So that's the reason why we are not cutting lines. But having said that, when the volunteering opportunity came, when Pfizer was looking for people, it had careful idea of who they wanted to be, and they selected the volunteers accordingly. So a lot of us never made it to that list. But even during the clinical trials, I would feel confident to undergo a clinical trial and have a vaccination.
So was the contribution a global thing, or was it just that everyone was on their own and they were just trying to grab the prize?
Oh, it was because of a global effort. Because when you come up with, when you identify what a coronavirus is, and when you know what a structure is, when you look at its component, quite often you don't have to share those details with the rest of the companies, or quite often you don't have to share any details with other universities. What happened in coronavirus is that it was a global response. Everything that was discovered in the early phase was openly shared by universities and companies. And in fact, most companies that are now making a vaccine have signed a pledge saying that we're not going to make a lot of money out of this. And in fact, companies such as AstraZeneca has openly said that we will not make any profit from making vaccines. So this has been a little different response compared to previous vaccination programs, which run for profit.
Okay, so we have a question. Why is the World Health Organization not more convenient now in recent times as it was in the beginning?
I'm not sure if I can answer that. I mean, WHO can suggest a lot of people, but the problem with WHO is that it doesn't have any regulatory power, meaning it cannot just enter a country and say, hey, just do this. It doesn't have any power. So in the past, it used to advise and suggest people. And when this coronavirus became a pandemic, it was the responsibility of WHO to decide and officially call it as a pandemic. And that's why you heard from WHO more often in the early days. But once they declared it as a pandemic, and once they suggested how to deal with it, the rest of it was just left to the countries to actually manage it, make sure they follow those regulations, and then the rest was left with coming up with a vaccine. And WHO has no responsibility on that. So that's probably the reason. And they're the only one who can call something as a pandemic. And that's probably the reason why you heard them so often in the early days.
Okay, we have a very interesting question now. How are pharmaceutical companies addressing the problem of rising anti-vax culture?
That has been a very old news for pharmaceutical companies. We run a lot of social campaigns. We run a lot of advertisements. And as employees, we go around and volunteer in different festivals and explain what vaccines do. So there's a lot of proactive work that companies do. But more than the companies, the government actually runs a lot of proactive organization. The anti-vax culture seems to be a bigger issue because it's very easy to now share your opinion on Twitter or social networks. So in the past, if you say you didn't like the vaccine, two people around you would have heard it. But now if you say you don't like the vaccine, an entire planet can know about it because of the social media interactions. So the threat is real, but keeping up with the threat, the government and the companies have done a lot to go against it.
Okay, this is going to be the last question, and it's from me. So in your presentation, you said that our body has a database of sorts for all the diseases or whatever we get. So when we have that and when we overcome it, why do we still say catch a common cold or get a ear infection or a throat infection?
Because that's the new virus.
Sorry?
Because that's a new virus.
A common. So every time we get a new common cold, it's a different virus.
Yes.
Oh, okay.
Yeah. Every common cold is a new virus.
Okay. That's good to know. And on that note, we will be ending this talk.
We have another
Professor
question. We have another question. Could you highlight the difference between Sputnik 5 and the Pfizer vaccine? Like how a comparative relation?
I haven't looked into the scientific research from Sputnik. I believe it's not open yet, or it's not released to the public yet. But I can't speak for it. But as I mentioned before, if any vaccine that's effective over 70%, it's often a good candidate. But if its efficiency is beyond 90 or 95% or 99%, then it's wonderful. So if Sputnik 5 comes up with higher vaccine efficiency compared to other existing vaccine, then I'll be the first to take it. So it all comes down to the kind of data they produce and release it to the world. But essentially, you could take any vaccine as long as it is effective.
If we don't have any more questions from the audience, we would. Yes, we have one more. If one gets vaccinated, is there any chance of getting affected by the virus again?
No. So it's highly unlikely, but there's a chance that you will feel sick for a day or two. But because your body has been trained with the vaccine before, so if you get affected by COVID in the future, your body will immediately jump on it because of its previous engagement with the vaccine. So quite often you will not fall sick. You may know that you're sick, but that's for like half a day, 1 day or 2 days. But you will certainly not go to a point of being hospitalized.
Okay. So if anybody else has any other question, they are free to, unmute and ask sir directly. We'll wait for say a minute or two.
Okay, I'm very impressed with the engagement. I really thank all of you for asking important and wonderful questions. I really appreciate your engagement.
Sure, everyone is thankful for you to taking the time and answering the questions. So more than that.
No problem. It's nice to chat with everyone.
Yeah. Okay, I think we can end the Q&A session on that.
Okay, cool.
We'll now have Professor Goribal delivering the word of thanks for the talk. Professor?
Hello. Is it audible?
Yeah, I can hear you. Yeah.
Good morning to everybody.
Yeah.
It's a great privilege to me.
Good morning to everybody.
On this occasion.
Privilege to me.
With gratitude.
On this, my gratitude, Dr. Giraddi, senior scientist.
Sir, I'm so sorry to interrupt, but your voice is very flaky.
Maybe network. Hello.
You seem to be logged in with two accounts.
Maybe
So
network. Hello.
yes, sir.
Yeah, we can hear you now.
Yeah, now it's better.
Now it's okay. Uh, Good morning, everyone gathered here. It's a great privilege for me to present my vote of thanks on this occasion. First of all, I would like to thank Dr. Rajshekhar Giraddi, a senior scientist, Pfizer, USA, for spending his valuable time in presenting a wonderful talk on virus classification, how like yellow bottom and top. Really, it was a very and thank for your invitation. At the same time, I would like to manage MSIT, AT, and the Angels for their this wonderful webinar. Extend my job and
We can't hear you.
I also thank in my of AI and hello.
Yes.
Head of the department for their support and execution of this particular event. And also I thank our head of the department, Dr. Putragowda, HOD Computer Science, ATME College of Engineering, Mysore, for their full support and execution for this particular event. I also thank each and everyone who supported, who attended this event across the globe and made the day wonderful. And I thank students community and the faculty community and the various people who attended and made the day wonderful. And I thank the organizers of both institutes, faculty members, and students once again for their wonderful support and execution of this event. Thank you, everyone. Have a great time. That's all. Sign off from my side. Thank you very much.
Thank you. Thank you for giving that. Yeah.
Once again, I would like to thank everyone who attended this talk. I have shared a feedback form link on chat for both YouTube and the Google Meet. So please take 2 minutes of your time to fill out that form. It's just for our data collection. And that's about it. Thank you for your time. Have a nice day.
Thank you. Have a nice day. Bye for now.
Thank you, Dr. Raj.
Hello.
Thank you. Thank you. Thank you.
Yeah, hi.
Hello.
Yeah, hi.
Hello.
I'm still here.
Thank you, Rajshekhar, and we would look forward to many more interactions, which are so interesting. We are getting so many feedbacks that it was very informative, very enlightened to be a part of this webinar. This is what the feedback that we have right now from the participants. So looking forward for many more interactions in the coming days.
I'm happy to hear. Yeah, and happy to talk in the future.
Yeah, thank you for your valuable time that you have spent and enlightened this day for us with this talk. Thank you.
Yeah, thank you for inviting me. I hope you all have a wonderful day. I'm going to disconnect the call now. Yeah, go ahead.
Yeah, thank you.
And on a lighter side note, for all the students present here, please go through the Brainium's website to check out the fest coming up from tomorrow and for tomorrow and day after. Thank you. Have a nice day.
Hello. Wait, let all of them work. See.